All right, within this review, we're going to cover the first 3 weeks in hematology 2. So we will be talking about hemoglobinopathy, thalassemias, different disorders of the white blood cells, such as Palgar-Hewitt, Chiari-Higashi, those kind of things, and so on. So we're going to get started here with chapter 26 out of your heme book, which is hemoglobinopathies. We're going to skip down to the slide called Overview. Basically, a hemoglobinopathy, the word itself just means a disease state involving hemoglobin. If you think back to med term, PASI refers to disease, and then the word hemoglobin. So it's a disease of the hemoglobin. So we have two categories here, basically. We have qualitative versus quantitative hemoglobinopathies. Qualitative involves that in which something about the actual globin chains is wrong. In other words, the amino acid sequence that makes up those globin chains has been altered. So something about it has been defective. In quantitative, it's just a quantitative lack. We can't make enough of the globin chains. We better know the quantitative as thalassemias. And so that's what you're going to hear them called from now on out. So whenever we refer to hemoglobinopathies, we're talking about the qualitative amino acid substitutions. When we talk about thalassemias, we are talking about quantitative lack of globin chains, just not enough being made. And the next chapter 27, we'll talk all about thalassemias. So just a reminder, the structure of the globin genes here, there's 6 globin genes on 2 different chromosomes. You don't need to worry about the chromosome numbers or anything like that. And again, a reminder of this chart that comes back from Hematology 1, in which it shows you all the different types of hemoglobins that are possible and the time periods that they occur. In adults, we have, again, hemoglobin F, A2, and A. So in hemoglobinopathies, there can be a number of genetic mutations that puts them into having something wrong with their globin chain. Point mutation is probably the most common type of genetic mutation. So the next couple slides just cover that. I'm not going to go into detail on this. This is not something that out of everything we need to cover, I'm just going to skim through those. All right. So most clinical abnormalities with hemoglobinopathies do refer to the beta chain. And the most frequent abnormal hemoglobin we find is hemoglobin S, or what you better know as sickle cell anemia. Okay, waiting for the next slide to click here. All right, so there is classification of these hemoglobins. So you see the 4 classes here. We'll go through each of these classes. We're going to start, though, with our biggest hemoglobinopathy, sickle cell anemia, involving that hemoglobin S. A quick note, people that do have sickle cell anemia oftentimes are able to resist the effects of Plasmodium falciparum, which is a malaria. And it kind of makes sense. This is kind of a, if you will, a general evolution, as the people who tend to have sickle cell anemia are primarily in the malarial belt, so they're constantly being exposed to malaria. Sickle cell anemia, the reason that we get hemoglobin S, it's totally not a normal hemoglobin to have. What has happened is there has been an amino acid substitution. So you must remember that glutamic acid is replaced by valine, and that is how we become hemoglobin S. You can either be homozygous or heterozygous, just depending on your heritage. And so when that amino acid substitution occurs, and we have the hemoglobin S formed, it's very sensitive to how much oxygen is carrying, and that's why you get the sickle cell part. So whenever their oxygen in that hemoglobin decreases to less than 85%, then it will sickle cell up. And if you see here, it's a small picture, but it looks just like that crescent or banana shape there. Let's keep moving. So there's 2 types of sickle cells, the reversible ones that can just change back and forth depending on the amount of oxygen it carries, and then the irreversible that just always stay sickled in look. And the hallmark feature of sickle cell anemia is going to be vasoocclusion. Because of those sickle cells, those crescent-shaped cells, they're not deformable. They're not going to be able to traverse small spaces. And so what's going to happen is they're going to occlude and block, your blood vessels, and you're going to get a lot of pain with that as a result. So that's where the vasoocclusion comes in. A lot of people who have sickle cell anemia do get hospitalized when they're experiencing this vasoocclusion because it is very painful to them. So the lab diagnosis for sickle cell anemia, of course, we're going to see sickle cells. Target cells are a very key sign here as well. Nucleated red cells, basophilic stippling, Pappenheimer bodies, Howell-Jolly bodies, all can be seen in sickle cell anemia. It is a hemolytic anemia because of the blocking can do in the blood vessels. As the red cells try to get through there, it's going to shred them, and it's going to result in a hemolysis, a destruction of the red cells. That's the reason that you're going to see nucleated red cells coming out, because the bone marrow is going to try to compensate and replace those cells that got destroyed. Again, in bold here, it says sickle cells and target cells are hallmarks of the disease as far as under the blood smear. So definitely make sure that you make note of both of those. And target cells are actually a key sign in hemoglobinopathies, other ones besides hemoglobin S, and also in thalassemia. So they're a big sign in both of them. We are going to see increased reticulocytes as well because, again, bone marrow is trying to compensate. The screening test, there will be a solubility test performed. Hemoglobin S is going to be positive for that screening test, for the solubility test. Then we can go on and confirm it, and the way that we'll confirm it is through electrophoresis. So again, remember, electrophoresis is taking the hemoglobin, which is a protein, and migrating it on a charged field, and it will separate out into what makes it up. So that's how we can determine what types of hemoglobin somebody has. Us as normal, healthy adults will have A, A2, and F present. People who might have sickle cell anemia are going to have hemoglobin S showing up when there shouldn't be. If they are homozygous, they're going to see greater than 80% of their hemoglobin is going to be hemoglobin S. If they have what we call the sickle cell trait, they are going to be 40% S and the majority A still. So in sickle cell trait, it's really an asymptomatic thing. They don't have the vaso-occlusion or anything. They may have a few target cells, but usually their blood smear is pretty normal. And so it's just a trait. They're a carrier, if you will. So they can function normally because they still have mostly normal hemoglobin A. But if they do that electrophoresis, you still will see 40% of hemoglobin S showing up there. And again, that hemoglobin solubility test is still positive. So just a key here, hemoglobin solubility screening tests are always positive whenever hemoglobin S is present. Let's go to our next major hemoglobinopathy, which is hemoglobin C, found almost exclusively in African Americans. Again, another amino acid substitution that has occurred that you should make note of, and it's lysine is substituted for glutamic acid. A key feature here will be the hemoglobin C crystals, which is a short, thick crystal that we can find within the red cells. Again, lots of target cells because they are common finding in hemoglobinopathies. A negative hemoglobin solubility test, because like I said before, hemoglobin S will create the positive hemoglobin solubility test. In hemoglobin C disease, it does not involve S, so it won't be positive. And we can perform, again, hemoglobin electrophoresis to see the presence of hemoglobin C there. And so this picture is showing that hemoglobin C crystal, that short, thick, rod crystal that can appear in the red cells. There's another type of hemoglobinopathy that's a combination called hemoglobin C Harlem, otherwise known as hemoglobin C Georgetown, and we have a double amino acid substitution occurring. One is valine for glutamic acid, the exact same substitution that occurs in hemoglobin S. The other one is aspartic acid for asparagine, and I might have pronounced that wrong. But anyway, we can again perform hemoglobin electrophoresis, see the double here, see that hemoglobin C. This will have a positive solubility test. Now, I had said whenever you have hemoglobin S, you're going to have a positive solubility test. Well, there's not exactly hemoglobin S here. This is a combination, but because it has the exact same amino acid substitution as hemoglobin S, that's why the solubility test has come up positive. Another hemoglobinopathy is hemoglobin E. Again, negative hemoglobin solubility test. This one is dominantly found in Southeast Asia. There's hemoglobin O Arab. Lysine for glutamic acid is the amino acid substitution found in Kenya, Israel, Egypt, and so on. Again, many target cells because it is still a hemoglobinopathy. Then we can keep going, and we do have hemoglobin D and G. As you can see here, as we go through, I know I'm starting to skim because some of these get to be minor. Tons of hemoglobinopathies, over 800 have been found. I do want to take a quick look at this one. This is a combination of a hemoglobin S and hemoglobin C disease. It's going to kind of resemble sickle cell anemia because of the hemoglobin S now present again. So you're going to see both hemoglobin S and C on the electrophoresis. You will still have a positive hemoglobin solubility because hemoglobin S is present again. Another combo effect that you can get is hemoglobin S with a beta thalassemia. We can have hemoglobin M, which is met hemoglobin. Remember, met hemoglobin is when your iron is in the 3+ state. It normally should be in the 2+ state, but in met hemoglobin, it's in the 3+ state. And what happens with met hemoglobin is it doesn't want to deliver oxygen. It really likes the oxygen, just wants to hang on to it. As a result, if it can't deliver the oxygen, your tissues are going to have cyanosis and death. So there are some other unstable hemoglobin variants that we can occur. The most prevalent one is hemoglobin Colne, and then the others are Zurich, Hammersmith, and so on. So you can read those there. There are hemoglobins that have increased oxygen affinity, meaning that they really, really like their oxygen and don't want to let it go to be delivered. Things like that would be carboxyhemoglobin, which is due to carbon monoxide poisoning, and that met hemoglobin, again, that we talked about. And then hemoglobin Kansas would be an example of a hemoglobin with decreased oxygen affinity, meaning that it will quickly deliver that oxygen and release it. All right, so that is the chapter on hemoglobinopathies. The ones that you should mainly concentrate on are, of course, sickle cell anemia and sickle cell trait, hemoglobin C, and then just know a little tiny bit about the others, but those are your two big ones out of the chapter. We're now going to switch over into chapter 27. That was on thalassemias. So give me just a second for my PowerPoint to switch here. All right. So again, remember, thalassemias is all about a quantitative lack in their globin chains. They just can't produce enough of them due to a gene alteration. So as a result, we're going to see some anemia occurring here because we're not going to be able to make red blood cells. When we name the thalassemias, they're either named as an alpha thalassemia or a beta thalassemia, and they're named according to what globin chain got affected. So in alpha thalassemias, there's problems with the alpha globin chain not being enough. And in beta thalassemia, it's the beta globin chains. All right, so epidemiology, there is a thalassemia belt, so it's Mediterranean through Middle East, south through Africa. This seems to follow where malaria is present, so kind of similar to that sickle cell anemia, where it occurs quite frequently where malaria is prevalent. All right, so we are going to skip that slide. So pathophysiology. Thalassemia is a microcytic hypochromic anemia. There's just not enough hemoglobin to develop the red cell. They're lacking globin chains. So you also see a decreased red cell survival because of the lack in globin chains, which is what comes about with the anemia. So we're in beta thalassemia, you're going to see unpaired alpha chains, and they will precipitate and damage the red cell surface. And then as a result, we're going to see premature death. So that's going to lead to what we term ineffective erythropoiesis. And so ineffective erythropoiesis is just a term that's saying that the red cells couldn't be effectively made. There was something happening where they just couldn't get made properly. And in this case, we can see why, because of the precipitation and damaging that's occurring, and then we destroy those red cells because they're damaged. All right, so I am just kind of skipping here. I want to focus a little bit here on hemoglobin BART, which is 4 gamma chains, so you still need to know that. All the alpha genes are absent in hemoglobin BART. So absence of all 4 alpha genes. Hemoglobin BART, a fetus cannot survive with just hemoglobin BART. So it's a term that we call hydrops fetalis. Because hemoglobin BART has such a high affinity for oxygen, it likes it so much, it won't deliver it. The fetus won't get oxygen to the tissues, and unfortunately, that will result in death. All right, so here it's listing just some genetic defects that can occur to alter those genes to make it so that their globin chains won't get produced correctly. We're going to skip through that, and we're going to start here now with our beta thalassemias. So in the beta thalassemias, again, it's the beta chain that has become affected, and there's 3 types. We have beta thal minor, intermediate, and then beta thalassemia major is on the next slide. Beta thalassemia minor, it's a very minor anemia, 10 to 13 or 10 to 13 grams. Few target cells. Again, like I said on the last chapter, target cells are seen throughout the thalassemias. Beta thalassemia intermediate is between the two. It's about a hemoglobin of 7. And then beta thalassemia major is our homozygous state, our very severe anemia. They're very transfusion dependent, meaning constant transfusions. So their hemoglobin levels sit very low at 3 to 4 grams. We will see hepatosplenomegaly, jaundice, bone changes, extreme poikilocytosis, so target cells, teardrops, elliptocytes, and so on. We can also see nucleated red cells because it's trying to replace and compensate, and same with the reticulocytes. There is a disorder called hereditary persistence of fetal hemoglobin. This is where you see increased levels of hemoglobin F in adult life. Normally, we have very little hemoglobin F, about 1%, and here you can see 15 to 30 is quite high. So slight microcytic hypochromic red cells. All right, so a couple other thalassemias that have beta genes that are affected one is hemoglobin Lepore. I am not going to concentrate on the other, really. I want to jump over into alpha thalassemias. So we have the alpha thalassemias again, is the alpha chain is the problem. So we have 4 syndromes, the silent carrier, in which they just have one globin gene that was deleted. There's no abnormalities. They're just truly a carrier. There's alpha thalassemia trait. So very mild anemia, again, microhypo. We have hemoglobin H disease, which is important to know, caused by the presence of only one alpha gene. So with hemoglobin H, we are going to see unpaired beta chains. Moderate hemolytic anemia, target cells again. And then finally, our last one is that hydrops fetalis with the hemoglobin BART. They are missing all 4 alpha genes, and so they will have 4 gamma chains instead. And with hemoglobin BART, because it won't deliver oxygen, we can't have that baby survive. That fetus will survive about the 3rd trimester due to some other hemoglobins like Portland that will make up to deliver the oxygen. But once Portland goes away and hemoglobin BART is left, nothing will be able to deliver oxygen anymore, and then it will result in be incompatible with life. And there's just a list of a couple hemoglobinopathy thalassemia combos. All right, let's talk about lab findings. We are going to see, of course, in anemia, decreased hemoglobin hematocrit, very low MCV. They're microcytic hypochromic anemias. We could see increased RDW if they're not treated properly. In the bone marrow, we might see some erythroid hyperplasia, trying to replace cells and to keep up with the anemia. And again, electrophoresis is a very good tool here just so we can see the types of hemoglobins present and what's going on. Okay, and I think I'm about done with this. Yes, I'm done with this chapter. Let's keep moving on through the first few weeks of Hematology 2. So the next, what we're going to do is chapters 28 and 29 out of your hematology book. I called it week 2 review when I first made this PowerPoint, but it's chapters 28 and 29. So it's all about the white cells diseases here. So we're going to start with some neutrophil diseases. The first one is Pelger-Hewitt. Again, Pelger-Hewitt are those hyposegmented neutrophils. And what I mean by hyposegmented is that their lobes are just 2. They have 2 lobes to their neutrophil, and we can call that a pince-nez appearance. It's important to make sure that you can separate Pelger-Hewitt from pseudo-Pelger-Hewitt. The normal Pelger-Hewitt anomaly does not cause anybody any issues. Their neutrophils, even though there's only 2 lobes, they can still function very normally. So people with Pelger-Hewitt, they're okay. The pseudo-Pelger-Hewitt, though, is a concern because there's a lot of different diseases that can mimic Pelger-Hewitt, in which certain leukemias will have their neutrophils appear like they're Pelger-Hewitt with just the 2 lobes, but really these are going along with a quite serious condition like leukemia. So we want to make sure that we can differentiate if it's just Pelger-Hewitt or if it's something that's mimicking it and is quite more serious. Our next disease is Kagashi. The key feature here is large peroxidase positive lysosomes. We are going to see a fusion of granules, so you're going to see these big, large lysosomes inside the cytoplasm. Very increased susceptibility to infections, so their neutrophils do not function normally here. As a result, if the neutrophils can't function in phagocytized bacteria, they're going to be increased susceptibility to infections. They have thrombocytopenia, so you might see some hemorrhaging, and their platelets aren't functioning properly either, so that's why their bleeding time would be prolonged. For elder-Wiley, this is a decreased mucopolysaccharide degradation. In other words, they're not breaking down the mucopolysaccharide properly, so what we could call a mucopolysaccharidosis. In other words, they have just too much of that mucopolysaccharidosis or lipids in their cytoplasm. It's going to look like deep purple to lilac granules covering that neutrophil. And then the last one out of the four is meheglin. They're going to have doly body-like formations inside the cell and giant platelets and thrombocytopenia. Those are the three key features seen in meheglin. For macrophage diseases, we had two that are important, Gaucher and Niemann-Pick. For the Gaucher disease, the enzyme that's deficient is beta-glucocerebrosidase. So I always remember the G's go together. For the Niemann-Pick, the enzyme that's deficient is sphingomyelinase. The way students have remembered this in the past is that you pick your sphincter so that they can remember what enzyme is deficient with the disease. Both of these have very unique macrophage appearances. In the Gaucher disease, the macrophage will have what we call chicken scratch or crumpled tissue paper cytoplasm, whereas in the Niemann-Pick, it's going to be foamy cytoplasm. In other words, lots of vacuoles in that cytoplasm. So you could adjust that saying to pick your foamy sphincter if that helps you remember those features with it. For a monocyte, for our next disorder is infectious mononucleosis. This is caused by the virus Epstein-Barr. Lymphocytes are what is affected here. They are the primary cell that gets affected. They become reactive, so that's where we see those big variant or reactive lymphs. We diagnose this in a lab using a kit looking for the presence of heterophil antibody. So how can you tell if somebody has a bacterial versus a viral infection if you just look at their CBC? Well, here's a few key ways to do that. Bacterial infection is typically going to have an increased white count and increased neutrophil count. Again, neutrophil's primary function is to phagocytize bacteria. So when we see bacterial infection happening, we're going to increase our neutrophils to help kill those bacteria. In a viral infection, you can see a normal or decreased white count and increased lymphocytes. So there's a quick way, just by looking at a CBC, how you can start to begin to differentiate between the two. And also in bacterial infection, if it's a severe enough bacterial infection, you can see some other changes occurring under the blood smear for the neutrophils. Toxic granulation, doly bodies, and vacuoles inside those neutrophils. All three of these are very abnormal to have. They all go along with severe bacterial infection. Toxic granulation is just going to be heavy, intense granules that appear on those neutrophils. Doly bodies are going to be those round blue inclusions in the cytoplasm. They're made up of RNA. And then vacuoles. You should never have vacuoles in neutrophils. Monocytes are totally fine to have vacuoles inside of, but in neutrophils, that's a sign of a bacterial infection of the phagocytosis process occurring. Let's talk about some definitions that were in these chapters. All the L's here. Left shift is a term that's describing increased immature granulocyte count. So if you're looking at a smear and you're seeing a lot of band neutrophils, metamyelocytes, myelocytes, those are all a term to be used to describe that would be left shift. The term leukoerythroblastic, is a term used in malignancy, and we use this to describe immature white cells along with the presence of nucleated red cells. So if you look at the word leuko for the white, erythro for the red, and blastic for the immature part and nucleated part. And then our last L word was leukemoid reaction. A leukemoid reaction, again, is an exaggerated response to a bacterial infection. Typically, their white count is up around 50 or more. Also within these chapters 20 and 29, we learned how to differentiate between a relative and an absolute count, and how to calculate an absolute cell count. So again, remember, relative is referring to an estimate, a percentage. So if you have 78% neutrophils, it's saying, I estimate that's about 70% of your cells are neutrophils. It's a relative number. Whereas absolute is the actual number of cells. So they're going to say, well, no, you have 8.5 times 10 to the ninth liter of neutrophils. So you can see the difference where relative is a kind of an estimate and absolute is an actual number of it. To determine the absolute number, we take the white count times the relative number put back in its decimal format to get that absolute number. So if you had a white count of 10, as you see here, and you had 40% neutrophils, all you would have to do is go 10 times 0.4. So remember, just to put that percentage back into decimal form, and that will give you your absolute number. And then chapter 29 was an introduction into malignancies. There wasn't a heck of a lot they wanted you to take away from this chapter. I did summarize a few key points here. The difference, again, between leukemia and lymphoma, and I know we talk about this later when we get into those chapters, is the origination. Leukemia is bone marrow. Lymphoma is the lymph system. There was a mention in this chapter on Epstein-Barr virus and how it's been linked to causing Burkitt lymphoma. And then the Philadelphia chromosome. Later when we get into the leukemia chapter, we're going to review the chronic myelogenous leukemia, CML. And Philadelphia chromosome is a very big part of CML. It will become positive in it. What Philadelphia chromosome is a gene fusion between BCR and ABL, and it's a translocation of the chromosomes 9 and 22, so you still must remember that. All right, so we are going to go into our last two chapters on this portion of the review. These are going to be chapters 30 and 31 on basically cytochemical stains and cytogenetics. We're going to concentrate mostly on the stains. So the first question here is, why do we even use stains? The reason, one good reason would be blast cells. When you're looking at a blood smear and you're seeing a lot of immature cells, such as blast cells, you're not going to be able to tell if that blast cell is a pronormoblast, a myeloblast, a lymphoblast, a monoblast, whatever. You just know it's a blast, and it looks immature, and it has nucleoli, and it's large and dark. So if you look here, you can't tell the difference between these two pictures. There are two different types of blasts, but you have no idea which type they are. So that's one reason to use stains, is to help differentiate what type of blast am I looking for, what type of leukemia might be occurring here. So I have listed the stains on the next two slides in a table, what they're used for, what are they stain, and what we look at. The first two rows, myeloperoxidase and Sudan black B, are used to help determine AML from ALL. AML will be positive, ALL will be negative. So the myeloblasts in AML will become positive in both myeloperoxidase and Sudan black B. Myeloperoxidase stain is going to stain exactly what it's titled, the myeloperoxidase enzyme. That enzyme is found in the primary granules of neutrophils. In Sudan black B, it is specifically staining lipids in the granules of neutrophils. In both, we are looking at the myeloblasts, the blast cells, to determine if they're positive or negative. So remember that myeloperoxidase Sudan black B, AML positive, ALL negative. Chloroacetate esterase, and then the other esterases are the next two rows. Chloroacetate esterase is what we call our specific esterase, and then we have the alpha-naphthylbutyrate and alpha-naphthol acetate, which are non-specific esterases. In the chloroacetate specific esterase, we are going to see myelocytic cells be positive. In the non-specific esterases, monocytic cells are positive, so make sure you differentiate there. The last three stains, PAS is periodic acid shift. ALL will be positive, and only the erythroid type of AML is positive. All the other AMLs are negative for this, so make sure you know that. It specifically targets glycogen in the cells. LAP, leukocyte alkaline phosphatase stain, this is the one in which you had to score and count it. I'll talk about that here in a minute. But this is to help differentiate between a leukemia reaction and CML, chronic myelogenous leukemia. So with these two diseases, both have an extremely high white count. We just talked about leukemia reaction was an exaggerated response to an infection. It has a very high white count. Well, so does CML. CML leukemia has a high white count. So this stain will help differentiate between those two. Um, in this one, this is one of the few that we're looking at banded segment and neutrophils. Most of the other stains we're actually looking at the blast cells. And then our last stain is TRAP, tartrate-resistant acid phosphatase. We specifically use this to diagnose hairy cell leukemia. It is isoenzyme 5 is resistant to this stain. So those hairy cells have tons of isoenzyme 5, so they will be resistant to the stain. So we're looking at those lymphocytes, those hairy cells. Hairy cells, again, are lymphocytes with hairy extended projections from their cytoplasm. So a good saying to remember with this is trap your hairy monster. All right, so let's go into the lab score because you got to remember how to score the stain. So we are going to look at 100 banded segmented neutrophils, and we're going to score them from 0 to 4. A score of 0 would be that they didn't react at all with the stain. A score of 4 is that they extremely, very intense color reaction. We're then going to multiply the score by the number of cells that occurred at it. So in the example here, we had 20 cells that were a score of 0. So 0 times 20 is 0. We had 45 cells that were a score of 1. So that'd be 45. 25 cells that scored 2. So that'd be 50. 3 times 5 is 15, and so on. Once you're done multiplying, then you add that last column. So 0 plus 45 plus 50 plus 15, and so on. So we get our lab score of 130. So a normal reference range is anywhere between 20 to 100. So that example that we just used, which was 130, that would be an increased score. Different diseases that are going to cause an increased score is leukemia reaction, polycythemia vera, and pregnancy. Decreased lab scores are going to be seen with CML, paroxysmal nocturnal hemoglobinuria, myelodysplastic disorders, and sideroblastic anemia. But you can very much see why we can differentiate leukemoid from CML with the stain. Leukemoid has a high score, CML has a low score. And then chapter 31 was cytogenetics. Cytogenetics is just the study of chromosome abnormalities. I don't go a lot into this chapter because I feel like it's pushing a deeper, like 4-year level. There's just 2 that I want you to note here. CML, again, that Philadelphia chromosome translocation between 9 and 22. And then acute promyelocytic leukemia, you should also know the chromosome translocation that's occurred here, which is 15 and 17. All right, so that is the end of the first 3 weeks of Heme 2 review.